So let's dig into this paper. So this is kind of his first Alzheimer's work in a way. And I'm curious who the last author here is, Richard Schenck. And I would be a bad investor if I didn't look into Richard Schenck as well. Well, I will not be talking to Dr. Schenck anytime soon. He is dead, passed away. He's from Bluebell. I actually know that town. So perfectly fine paper. Not going to go into the details right now. I'll do that offline. But I do think that one thing from my experience is the drug industry has tried to drug the nicotinic receptors over and over and over again. And just by happenstance, I bought the first ever nicotinic drug called methamphetamine, which at the time was approved for hypertension. And there was a company that actually spun out of R.J. Reynolds, the tobacco company, and they called themselves 𝗪𝗏𝗏. And they actually took this drug, methamphetamine, they took an isomer of it and tried to develop it for basically anything they could. They did every CNS disease possible, including Alzheimer's, ADHD, and they sort of settled on depression as their main target. They failed. They did not find any effect in virtually any of those diseases. I think that almost every drug company at one point or another has had an alpha-7 specific antagonist. Now, just because they've blocked alpha-7 doesn't mean you block the receptor A-beta interaction. But I will say that if alpha-7 was this big player in Alzheimer's, I think we would have heard a lot more about it, and there just isn't much talking about that. Now, whether or not every research paper somebody publishes is right, or that's not the point. Very few scientists even bat 10% on these things. But I do think this alpha-7 thing comes up a few times, and I can't speak with the authority of somebody who's spent their whole life on Alzheimer's specifically, but I will say I think I know Alzheimer's, and I think that there's not much going on in the alpha-7 world. One of my friends has an Alzheimer's biotech startup. I'll actually make a note to call him. But I don't think alpha-7, the nicotinic receptors, the only possible explanation there is that, acetylcholine signaling is implicated in Alzheimer's because the first ever Alzheimer's drug was an acetylcholinesterase inhibitor that degrades acetylcholine. So, there is some link there. But the alpha-7 receptor is just one of, there's two kinds of nicotinic receptors. There's two kinds of acetylcholine receptors. There's nicotinic and muscarinic. And just of the nicotinic, there's several subgroups. I think there's like 5 or something. So they form these ion channels, and they're sort of like, they almost look like a flower, and each petal of the flower can be a different sort of, each petal is a different isoform of these, I guess, units, these subunits. Yeah. So you have alpha-7, alpha-4, alpha-2, etc. So anyway, the sort of nicotinic space has been a little bit not great. And by the way, that first Alzheimer's drug did not work very well. The 𝗪𝗏𝗃 is generally thought to be kind of a bit of a dud. Although in a disease without any drugs, even a dud is welcome. Filament is one of the biggest proteins in the body. It's like 2,500 amino acids.
And it's everywhere.
Yeah, it's not a surprise.
It's in almost every cell.
Yes. And so it's not
overexpressed in some places, and it happens to overexpress in Alzheimer's.
Well, I won't take that for granted just yet, but here's what I'd say. Filament A, or filament in general, it's not surprising to me that this highly reactive amyloid, literally it's amyloid beta, amyloids are like literally the self-aggregating drugs, and they're very promiscuous. They have electrostatic profile that binds them to everything. And I think the prevailing theory in Alzheimer's is not this one. It's this idea that the self-aggregation of amyloid beta kind of creates this very strong immune response that ends up, as all immune responses do, kind of destroying gray matter or destroying neuronal integrity and synapses and things like that. I don't think
and it's binding somewhere, and that's causing them to accumulate.
Well, the alpha A beta 42 will self-aggregate, right? That's what the word.
Yeah, so with its,
Yeah, that's what the.
Not at a point of a receptor, you're saying.
Exactly. So 𝗂𝗡𝗏42 self-aggregates into filaments and other like higher order structures. And even those higher order structures have their own affinities and so forth. And there's a lot of work on this from donanemab at Lilly, where, depending on the A-beta and even depending on post-translational effects of the A-beta, you can have different events. Like there's pyrophosphatase, or there's other like small modifications that could be made to the peptide that changes it. And there's a lot of theories that have been promulgated on this. As you know, I mean, there's 100 theories about why Alzheimer's happens. To me, it's not really plausible that the alpha-7 receptor is relevant here. I think you could actually knock out alpha-7 completely and still, I'm going to look into that, like the classic mouse knockout experiment.
The mouse model.
Yeah.
Yeah.
And it's not hard. Like you and I could probably buy alpha-7 knockout mice for like $500 and do our own experiment, commission our own experiment if we had to. If we really start beefing, maybe we'll do that. I don't think that you could have such an impact just from, Alzheimer's is a catastrophic process, and you're really just knocking alpha-7 down. Alpha-7 is a self-contained ion channel, right? So like, I'm not sure what downstream there is there, but like, of course there is something downstream, it's signaling somewhere. But like, it just doesn't seem like if you disrupted alpha-7 integrity or you inhibited it or caused it to be down-regulated, I'm not sure what the thesis is, but regardless, like, to me, it doesn't mean a lot that A-beta binds to something, and then it doesn't mean a lot that it's sort of filament and alpha-7. Now, stranger things have happened, right? If you would have told me that, certain mechanisms were relevant for certain diseases, many times I would say, no way. And the results come out and you're like, oh, actually, that was right. And so that, it's always reading tea leaves kind of here. The other thing,
this one has a similar mechanism.
well, I just want to say the other thing that's really crucial here is from the medicinal chemistry side, PPIs, protein-protein interactions, which is what we're talking about here, right? Mm-hmm. PPIs are some of the hardest things to engineer. Like, this was serendipity. And again, I'm maybe being generous. There are probably some fellow short sellers out there who are like rolling their eyes right now. But like, it's very hard to design a drug that interferes with the protein-protein binding of two separate proteins, right? Typically in pharma, what we're doing is we have an X-ray crystal structure of a specific region, of a specific protein, and we're making this hydrogen bond network to that specific region. And usually it's a receptor pocket or it's another sort of pocket on a protein. I, for one, cannot find the filament A complete structure. I have like these partial structures. So I'm going to look for a crystal structure for filament A. It may not be possible to create the entire filament A crystal, but thanks to AlphaFold 3, you can actually you can actually render them with some decent fidelity with AlphaFold. So
he tested it initially, I think, with a peptide, which is in one of the papers. It's 𝗂𝗡𝗏 something. I think that's where he kind of used to narrow down where it was going to bind.
Yeah, and I think it's not surprising that like a very hydrophobic, set of residues on a huge protein would also, bind to another set of very hydrophobic kind of region of A-beta, for example. So like it's like, to me, the fact that there is something does not mean that there is something that is working. And, you have to elegantly kind of tie, the preclinical science to some kind of phase 1 PK type science to some phase 2 clinical science. And to me, that story is missing. There isn't this like connection. Now, I mentioned before that I met Remi in 2004. I was an analyst at a firm that his college roommate ran, a major hedge fund. And I was a big fan of Remi. I like Remi a lot. He's a kind of like do-it-yourself entrepreneur. He is not a scientist particularly himself. He went to Oberlin with my old boss, who I still regard very highly, and I think vice versa. I was very disappointed and upset that Remi resigned, because Remi this is his life work. This company is his life's work. Pain therapeutics almost became very successful, and the fact that he had left the company, or God knows what happened exactly. I wasn't in the boardroom. Maybe you should be on the board, by the way. The fact that he left without an acknowledgement from the company or anything. This is a company that he started 25 years ago, worked day and night, and he's 3 months away possibly from the results of his life's work, or at least the latest chapter of his life's work. You know, to me, that is sort of, one, of course, disappointing. I feel terrible for the guy, but it's also very alarming because, you know, if it was just a case of, hey, you know, there's a lot of FUD. I hate that term, by the way. There's a lot of FUD, and you know, we'll see the result when we see the result. There are many companies that have been in this situation. You just stand tall and say, look, we're not going to comment further until the results come out. Yeah, my experience is if they're going to take Dr. Wang to trial, they need witnesses. And the witnesses are Remi and Lindsay. And usually what ends up happening, look at the...
He worked with people in the lab a lot closer.
Well, they're going to put them all on the stand. And in the SBF trial, Sam Bankman-Fried unfortunately had to confront every one of his co-founders and major senior colleagues, except for one, who I should mention, but almost every ex-employee. And some of them he went to college with, some of them he had a long history with, one of them he was romantic with. And it's not uncommon, I doubt any of those people wanted to be there, by the way. It's not uncommon that they simply say things, and I know this from my own case, where they say, oh, are you going to testify against him? Here's the script you have to read. Oh, you don't want to do that? Cool, you're his co-defendant now.
Here's your charge.
Yeah, good luck in prison. And you know.
And by the way, your brother's been doing some illegal
You're right.
activities too.
Right.
We're going to have to check it out.
Absolutely. So look, there's nobody that hates the DOJ more than me, and I'd be, you know, just as soon on Remi's side as an individual, but maybe not on his side as this is the great drug for Alzheimer's. So one of the things that every short seller worries about is that when a company has disappointing results, that they try to spin them or delay them or try to avoid reporting them, or even
do another trial, like plan another trial.
Sure, and another trial is fine because I mean that's fine because like at the end of the day, Wall Street understands like that means it failed. But you know, it's always in the back of a short seller's mind that like even if I'm right and the drug doesn't work, what if they say it did? And I think that at least for the short seller side of things, with the DOJ and the SEC, it almost to some extent like running this company, like they're going to be very transparent about what this trial result was. I don't think there's going to be any obfuscation or any spin. It's just going to be like, here's the p-value, here's the result, here's the result of the primary endpoint. But as you know, even though trials are cut and dry, or they should be, there's things like intent to treat, modified intent to treat, last observation carried forward, best observation carried forward, and you can have like these little discrepancies. But typically a study fails or doesn't. I just want to respond to somebody who says high energy reviews and investments. Martin has been paid by the shorts. Okay, so first of all, I am the short. Nobody needs to pay me. I can.
Well, I thought you said you weren't short.
I'm not. But
you're considering.
Yeah, I'm considering it. And if I do short, there's no short seller who could pay me enough money to buy my opinion. My opinion, the last time I shorted a stock, I made $20 million called Vital Therapies, and I wrote a 40-page paper about it. Before that, I made a similar amount of money in Celadon. I mean, you can look at my track record of public shorts. It's very good in biopharma only. So, you know, I have no need to be paid by anybody. And nor would that be how I would make my money. It's also probably illegal for what it's worth, to pay somebody to bash a stock. So, I think that
if they overtly say that, that's not what's.
I think you have to disclose if you're being paid.
Yeah.
I'm pretty sure. But
lots of ways to pay somebody.
Yeah, it's nothing that I would even remotely approach because at the end of the day,
I'm not accusing you of that.
No, I know. Yeah. But here's what I propose: an intelligent way to, other than me pleading with you that is not valid. Here's maybe a way that maybe you could see where my point is. There are people, would you agree, in biopharma who've made a lot of money investing. They're very good at what they do. Would you say that those people have a skill or a talent? There's people sitting on billions of dollars who have analyzed drugs, and they know, and I'm not talking about Cassava, they know that the perils of open label analysis, and that.
No, there's definitely perils.
Okay, I'm glad you.
𝗕𝗏𝗏𝗃 was the big 400 patient blind placebo study. Totally agree.
Right, and all I'd say is that if you went to some of those people, I can think of a couple, 𝗕𝗍𝗓𝗓, Perceptive, one of the biggest funds, the Baker brothers. You got a good network, you're a wealthy guy. It would not be impossible for you to meet with the Baker brothers or Joe, and all these people are billionaires basically. Steve Boyd, Armistice, Rod Wong, RTW, these are just the biggest biotech funds, OrbiMed, et cetera, et cetera. And every single one of them will tell you that when we study biopharma, we don't look at the open label data. We try to avoid it because it tends to just be noise. It tends to misdirect. It doesn't mean it's worthless, but I think you have to be really careful. Yeah.
It can be directional.
Even saying that is, I think, these, again, don't take it from me. Talk to the best of the best, and I think they're all going to tell you you can't rely on this stuff. You can rely on a randomized study, and let's look at that data. It was not statistically significant. That is not good. It didn't
CMS, it wasn't powered to be.
Wait a second. I don't know if that's.
It literally wasn't.
Okay, but even if it was powered to be, and the p-value is 0.001, the change between drug and placebo is very small. That's why it's not
because it's a withdrawal study, and if the medicine is working and it's disease modifying, you will have a slower withdrawal than if it is working and it was just symptomatic.
I suppose I can buy that, but I wonder why they would do the study this way then.
I don't know.
You could do the study
I don't. I think I would have preferred, if you're going to stick and do open label, just to do the whole thing open label.
Agreed.
I mean, I don't think that it was particularly useful to do a CMS. And then they rolled those people that were on the CMS back into treatment, and so they have 24-month treatment data on Alzheimer's disease patients. Which, even if you study Alzheimer's disease, any placebo isn't going to perform like that.
I can show you plenty of placebo trials that did. 𝗕𝗏𝗏𝗏.
Not for Alzheimer's, not for 2 years.
𝗕𝗏𝗏𝗏, yeah, sure, absolutely. I've seen it. 𝗕𝗏𝗏𝗏, 𝗨𝗏𝗏𝗏𝗏, 𝗕𝗏𝗏𝗏. There's a host of these, and that's why we just don't trust open label data. It's not meaningful, and they were still declining in this study. They declined, and I'll look at the full data, but it's not like they got arrested development or they started reversing. Placebo declined almost as much as drug. I mean, it was basically the same thing. It was not statistically significant, but I don't even know. I can do a little biostatistics. I'm not even sure what sample size would get you stat sig here, given they didn't give the standard error, but I'm guessing the standard error is probably similar to other standard errors, and I mean, you're going to need, the Cohen's d value would be like 0.2 or something. Like, this is not a effect. And I understand what you're saying on the controlled withdrawal is possibly like one of these things that could putatively distort the rebound and so forth. But if you look at Vertex cystic fibrosis controlled withdrawal study, it is literally a straight line up, then a straight line down when you withdraw it, and a straight line back up when you give it. And I understand CF is not Alzheimer's. The brain is a really unusual thing.
It moves slower. It moves slower.
Yeah, but, you know, if it's going through acetylcholine, then it shouldn't. Acetylcholine is more of just like the tonic underpinning of the nervous system. This isn't like a learning molecule or like a synaptic integrity molecule. So like if you look at what 𝗪𝗪𝗃 does, 𝗪𝗪𝗃 works right away, take it away, you drop right away. It's like, it's not.
Yeah, well,
so if it works like acetylcholine, which is the hypothesis, it should actually be rapid on, rapid off. But
I don't think it's the hypothesis that it works like acetylcholine. I think.
Well, it's a nicotinic receptor, right? Alpha 7.
The hypothesis is that the neuron is being damaged by that interaction between amyloid beta and the receptors, and the neuron eventually dies.
But the alpha 7 receptor.
Yeah, but that's attached to a neuron.
Well, the neuron's got 50,000 receptors. I mean, it's not going to die because it doesn't have alpha 7.
It's the reaction down through the neuron. That's the proposed. That's the MOA, is that's actually causing downstream effects. It's not that it's shutting down that receptor. It's that it actually kind of kills the neuron through that receptor.
Yeah.
So it's not asymptomatic. It's.
Well, we hope so.
Yeah.
Yeah, I mean.
So I don't know. I would take a look at the one I just sent you, and then the 24-month data too. So you have 87 mild patients that stayed flat for 4 years, or for 2 years, 24 months on the drug.
Well, in 6 months, it lost 1 point. So flat is, it's not really flat, right, at 6 months.
Well, it's on the ones that went on placebo were down 1.
1.5.
And the ones that were in the mild group.
No, I mean, we look at all comers, right? We can't look at one group that did better.
Yeah, which one are you looking at?
The full analysis set.
Oh, I see. Like the mild versus moderate.
No, the whole thing.
The whole thing.
Yeah.
Yeah.
Yeah, if they knew that mild would be better than moderate, they would have done that ahead of time, right? This is just a like re-examination and saying throw out this data.
Most studies are done mild to early, and so the moderate segment group is a little bit of a risky thing to add to it. But that said, when I talk to the patients that have come off of these studies, there's a number of moderates that have improved radically.
I think I remember when Remi actually was doing an IBS study of naltrexone or naloxone. I forgot, it was ultra low dose naltrexone. And I talked to a bunch of patients as well, and they were doing great. This is about 20 years ago, and I learned a very painful lesson about patient testimonials. They're pretty much worthless. I've seen them, and even in cancer, I've seen them to be worthless. Like, it's just hard to, that
Alzheimer's though. Like there's
I don't think it makes a difference.
There's main symptoms. Like how does someone that is literally like losing balance and falling down, nothing else has changed in their life except entering a trial, they're diagnosed with Alzheimer's, that they stop losing their balance.
I would say the same thing about Duchenne muscular dystrophy of a dying boy. How could he all of a sudden start walking better in a clinical trial?
Yeah.
It happens. That when I trusted Remi on a subgroup analysis with patient testimonials last time, I lost millions of dollars. That was 20 years ago. I learned my lesson
the thing is,
many times over.
With this, there's about 7 families that I've talked to. Many of them describe very, very similar stories about what's happening. The main symptoms of Alzheimer's.
Is there selection bias though? Is there a selection bias there where you talk to the people doing well, but you're not talking to the people not doing well because they're drooling somewhere and not able to talk to you? So
100%. No, definitely. And because someone that's not responding to the drug isn't going to call me and say, hey, man,
right.
I would like to continue open label and they're terminating my program. That's how I ended up interacting.
Sure. You're comparing a probability distribution versus another probability distribution. And
definitely.
If you're talking to the right tail of one, you could be talking to the right tail of the other too, and you'll get the same testimonials. It's the mean and the median and the standard deviation that is relevant.
But in Alzheimer's disease, it's not the way the disease functions.
Well, you still I would just argue at 6 months,
There's not responders.
the data that I have, which is not personal anecdote, but the data that they put out is the drug and the placebo had almost no difference.
Oh, you're just looking at the CMS study.
Yes, at 6 months.
Yeah, you've got to look at the first 12 months.
Is there any other control data?
There's no more blind placebo data.
So the only control data for this drug does not work. I mean,
the
to
only
me,
control data, it's not the mild group showed that
they should have just done mild then.
Yeah.
I mean, I don't know how you can throw out. It's like saying my team won the Super Bowl in the first half. Don't worry about the second half. You know, it just doesn't. But the heuristic just doesn't.
You know, there's 87 patients, and
small study.
You know, half of them are placebo, half of them are mild, half of them are moderate.
Yeah, no, I get that. I just
and the disease, the way this disease is studied, is they're always broken into early, mild, and moderate.
Well, it isn't. It isn't like the primary endpoint can be, for example, for donanemab, for 𝗕𝗏𝗄𝗧𝗏, they did. Let's look at the 𝗕𝗏𝗄𝗧𝗏 study real quick.
I think they used tau. They did mild. They only did early, and they used cohorts based on tau.
Many Alzheimer's drugs have tried mild to moderate. It's not like some.
There are some.
I mean, including this one, right? So it's not like this was unusual.
The two that were approved were just early to mild.
Yes, there's been this. You know, you want to treat earlier, you want to treat longer for amyloid beta antibodies. That may make sense for amyloid beta.
Time is important. Yeah, that's why the 24-month data is I think very interesting. Is time is as important as quantity.
Time controlled, I would agree, but without a control, I don't know.
Yeah, and that's what we get in the phase 3 study that reads out.
Oh yeah, that's where all the marbles are, right? I mean, that's the big money. You know, it'll either go up 50x or it'll go down 90%, most likely. One of the two.
Yeah, that's why it's a bad short, in my opinion.
Yeah, I disagree, of course. But you know, that's what makes markets, as they say.
It does. It's just your downside risk on it is very large.
If there was a chance that it works, I would agree. I just don't think there's a chance. I don't think there's a chance at all, to be frank. But we have different opinions.
Clinical trial sites have been very positive about it, which you can say what you want. That's anecdotal. The patient families that we've talked to, they describe radical turnarounds. There are super responders to this drug. There are. That doesn't happen in the other Alzheimer's trials. The other Alzheimer's trials, they don't talk about anybody improving. It's just slowing decline. This drug has shown that it can improve, in the clinic.
I think we're not going to convince each other of what is
definitely not.
What is convincing information? Convincing information to me
phase 3 readout
is no controlled results.
I wish we had that.
Well, then we do. It didn't work.
No, you don't. You have a 6-month withdrawal study. 6-month cognition data doesn't mean anything in Alzheimer's disease.
Well, why'd they do it then?
And that's a good question.
No, they did it because they had a hypothesis that it would work, and it didn't work. Like in science, yeah, absolutely. In science, you absolutely, come on, Matt. I've designed these trials myself. You a priori say, my hypothesis is X. You don't just randomly pick. You do a protocol. You write a SAP. The protocol here is available. And they say, we are designing this patient population to target this change in ADAS-Cog and this separation at 6 months. And this is how much power we have to see this much change.
It was informational. They did not design that study to show statistical significance.
There's a protocol here, right? Let me.
I don't know for the CMS. That wasn't the goal of the study.
Yeah, go ahead. Sorry.
If it did, honestly, I would have sold some of the stock when those results came out if it was statistically significant in that 6 months, because it would have showed that it was symptomatic and it was maybe more like Aricept. So the fact that it slowly declined, there was some separation, but not rapid separation. I look at it as a positive. That's what I would want to see.
I understand. I mean, since we're recorded.
I wish we had a longer study, and I've got to run soon because I got to go pick up my kids from school.
Implore you. I'm sure this will be cut and taped either way, whatever happens here. You're right or I'm right. I implore you to save your money. Do not waste your money here. You're going to lose the money. That's just my 2 cents. I know your opinion is you might make $1 billion in this trade or more. And you know, for your own sake, I hope you do. You sound like a nice guy. And
Alzheimer's patients, but yeah.
Yeah, no, I want Alzheimer's patients to win too.
And I'm serious, I'll shave my head with you. Maybe not together, but...
Yeah, I don't think I'm going to be losing these beautiful locks, but I think let me do some more homework and we maybe can have another discussion.
Yeah, I'm game.
Thank you so much, Matt.
Thanks for the talk. See you later.
You too, bye. So that's how you have a conversation with an adult.